We are close to keeping everybody on time so that we can get all of you in and out onto your homeward travels on time. So the next topic, somewhat audacious, which is to talk about genomics and drug reimbursement white paper from the Cleveland Forum 2013. It sounds like a great title of something, I'm not sure what. And so the question is, can you help us sort of think through, or can this panel help us think through, what might we take from this discussion that we've had about realistic objectives, both near and far term, that might lead us forward. And I've got two folks on the panel that have not had a chance to be up on this podium before, so I'd like to introduce Drs. Balwell and Wisha.
Hi, I'm Brian Balwell. I'm Associate Director of the Case Comprehensive Cancer Center and Chairman of the Taza Cancer Institute at the Cleveland Clinic. And I'm a bone marrow transplanter by trade.
I'm Max Wisha. I'm Director of the University of Michigan Comprehensive Cancer Center. I'm a medical oncologist with specialty in breast cancer.
Great. So, you know, we've brought a number of topics forward. And I thought we might want to address the issues that relate to application and reimbursement, both for the testing and for the drugs, and what, you know, if you will, what criteria do we want to be held to as being reasonable and legitimate for us to propose federal support for the things that we do, whether it be in the form of approval of our individual pathology labs, approval for tests, approval for drugs. So what are the sort of the key endpoints that we want to look forward to, and what processes, what methodologies might we be considering to allow for reimbursement? I'll just start out and remind us that CMS has dealt with this in the past. They have a functionality method for reimbursing testing and even drugs, where there's an effort to collect data and to use that in a thoughtful fashion. They did this for PET scanning in cancer patients, and they concluded after a couple of years of database collection that, in fact, PET scans were a very useful tool for clinical decision-making and treatment response in cancer. So that, if you could imagine a clinical trial with that endpoint, you might not understand what they were doing, but they were showing utility and value in a clinical setting of a test, which is their charge. So one possibility is some sort of a registry in that format, whatever that would look like. Another is the formulary that Richilski mentioned to us earlier. And we could hold ourselves accountable to a higher standard of everything being done in a clinical trial setting with validation that an individual set of tests or an approach to testing had clinical utility, certainly with approved drugs. So that's where we want to start, I think. And I'm going to open up to the panel. And maybe because Max and Brian haven't been on the podium, I'll start with you two.
Okay, well, I'm going to address a couple of issues that haven't been talked about that relate somewhat to these issues. And one is endpoints in clinical trials. You know, we've heard about drug approvals and how drug approvals are going to be also matched with molecular diagnostics. But I think also that there is some changing in the concepts of clinical endpoints that mean that we really need to think hard about the science of clinical trials and clinical endpoints because, one, the current endpoints using RECIST criteria and tumor regression haven't really been all that effective at increasing the longevity of our patients or leading to increased cures. Now, I think we have to applaud now the FDA for proposing some potentially alternative clinical endpoints, like in breast cancer, triple-negative disease, using complete pathologic response, because we know that is a good surrogate for ultimately decreased recurrence and actually more turns into more cures. I think we're going to need more biological endpoints. And what that means is a complete kind of rethinking of how are we going to utilize biological tests for endpoints, which by necessity in the earlier stages will be intermediate endpoints that then need to be validated in very careful clinical trials to demonstrate that those biological endpoints actually translate into improved outcome for patients. So I think those kind of endpoints need to be explored in the research setting, but hopefully then in the early research setting and involve some new technologies that are being developed. But then I hope that they will be translated into new drug development and ultimately drug approval.
I think one of our very large challenges is that medicine's changing. It's changing from a reimbursement based on volume model to a model in which reimbursement is based on value. And I think one of our challenges, one of our collective challenges, is to try to define the value of this topic. It's not something that we've done very well at historically, other than couch it in clinical value, although I don't know that we have a good sense of our current state with respect to cancer therapy across all disciplines in terms of the value we generate. But at the end of the day, new tests, new targeted therapies have to be demonstrated to show value, either clinically or financially, preferably both, for this field to advance. And so I think a couple of the points that have been raised during this program are very important. Number one is data collection. I would like to echo those who were at Asco a week ago and heard Dr. Shilsky's talk. It was outstanding. But I think that he brought up the idea of a national repository for data collection, and I think that's vital, because there's no way we're going to be able to show value to CMS, to payers, to whoever in this space unless we have data. Now, data's complicated because there's a lot of different platforms that have been raised in terms of what in fact are we going to collect. Secondly, I agree with Max totally. I think that we may need to start to look at, or more robustly look at, endpoints that have not necessarily been front and center within a clinical research space. But I think that we can't ignore this switch in medicine from volume to value, and we have to figure out how to be part of that.
Skip?
I guess I'm not positive that we can say that our current methods have not proven of any benefit. RESIST isn't the be-all and end-all, but it is the way we have developed drugs that have subsequently been tested in phase 3 trials and shown improvement in survival. And we collectively have assigned value to some of those drugs that provide a 3-month increase in median survival and cost $90,000 a year to give. So we have engaged in the process of defining endpoints, and we have assigned value. I think what we're really saying is we're not happy with that process, and I'm nervous about putting in place yet another process that develops sometimes less than completely objective measures of a biologic outcome, and then applying that to one of perhaps 3 settings for clinical outcome: registry, formulary, or a randomized clinical trial. I think, as Rich, I think, has suggested, we might be able to break the clinical trials paradigm a bit by looking at large numbers of patients, moderately well studied, with solid outcomes that we can measure as a way to decide whether something is useful or not. That's the way we get at the 97% of patients who aren't on clinical trial. I think the formulary idea has a lot of merit, and you might view that as the midway point between a simple registry and a formal randomized trial. And if resources were unlimited, I could see all of these things being developed. The problem we have is that resources are sorely limited, and exactly where we spend those resources, I think, is another challenge that we're going to have to come to grips with, either passively by watching it blow by us or actively by engaging.
Rich?
Well, let me just make one comment, and then if you like, Stan, I see Jillian found those slides that I sent, so I could just briefly review that so everybody's on the same page with what I have been talking about. But just one comment to Max's point. You know, it's oftentimes we hear criticism of FDA and other regulatory authorities that they are not sort of moving the ball down the field fast enough with respect to new endpoints and so on and so forth. And I mean, frankly, the burden is not on the FDA to do that. The burden is on us in the scientific community to develop the evidence that, you know, a new endpoint actually reflects a meaningful clinical outcome. And if we can do that, then the FDA is likely to embrace that. So I mean, so for you know, you could choose almost any endpoint you want. I mean, you know, there's a lot of research being done now on circulating tumor cells or circulating DNA, and you know, could a change from pre to post treatment in that sort of a biomarker be sufficient to, you know, provide convincing evidence of drug activity such that the drug might get some sort of provisional approval? Well, maybe, but you know, the FDA is not going to develop that evidence. It's up to the research community to develop that evidence and present it to the FDA, and you know, let them see if they feel that they can use it in meeting, you know, their sort of congressionally mandated requirements. So that's just a comment about that. If you give me 3 minutes, I'll show you a few slides here. So the proposed solution here is to the question of essentially how do you get the right drug to the right patient at the right time, and the idea is to create a what's called here a national pharmacy exchange, which would be a national formulary of targeted agents against common aberrations, then to create a registry of administered treatment and patient outcomes, and the participants would be patients, physicians, pharma, payers, and FDA. So how might it work? So here's the idea. We create this national formulary stocked with representative targeted agents against common mutations. Patients are registered in a national registry. The physician submits a validated test result from a certified lab and a drug request. The formulary reviews the request against some predetermined guidelines, and if it meets, you know, whatever those criteria are, the drug is dispensed to the patient. The doctor then treats the patient, and their outcomes are entered into a registry. And the next slide. So what's required to do this? Of course, well, pharma has to provide drugs. The patient has to agree to data collection. The physician has to submit the required follow-up data. And then the payers have to reimburse the treatment costs. This could look something like coverage with evidence development, which is what Stan was alluding to with the National Oncology Pet Registry. And then some third party, could be Asco, but could be any third party that has the interest in the resources, hosts the outcomes registry as an honest broker. So who benefits if we can set this up? Well, patients benefit because they receive the targeted agent that's matched to their molecular profile, and they have the opportunity to become what some people are now calling cancer information donors. Physicians receive guidance and treatment recommendations and access to drugs. Pharma receives data on drug use and outcomes to inform their research and development plans and life cycle management of their products. Payers receive data on drug use and outcomes to inform future coverage decisions, and regulators receive data on the extent and outcomes of off-label use, of drug use and test use, and also real-world safety data. So that's the basic concept. You know, as I was saying in the hallway, it only requires getting the drug companies, CMS, and the FDA to all buy into it, but you know, minor detail. And I think, you know, if people embrace the idea, you know, then hopefully collectively we can work toward making something like this happen.
So I'm wondering if I can't ask the panel to give us some thoughts on that. We'll start with Max.
So I think that it's really a terrific idea, but I think that actually the way we might look at this is a whole spectrum of how we approach this problem. This is a very important part of one end of the spectrum, which is the big end, actually. It involves most of the patients. As Rich said, 97% of the patients who are in on clinical trial. But I think it's also important that we then couple that with rethinking how the 3% that are on clinical trials, how we're really doing it. And I know the next session is going to address that in more detail, but just as it relates to this, I think that knowing, for instance, how patients become resistant to a targeted drug is just as important as knowing whether they'll respond to that targeted drug, since it's really been the experience with all targeted drugs, even talking about melanoma with BRAF mutations, that patients rapidly develop resistance. And usually the mechanism of that resistance is discernible by doing further sequencing and further genetic analysis. And I think as the tools evolve, such as the tools that Rich talked about with circulating tumor cells, that the need to do serial biopsies will become less as we can actually measure mutations in circulating cells, so that we'll be able to follow patients and be able to look at resistant clones developing as we give targeted therapies. Now, somehow, we're going to have to couple that kind of complex information coming out of the cancer centers and the research groups with the other 97% to understand also for these physicians, once a patient is on a therapy, let's assume it even works, which hopefully it will. When the patient becomes resistant, it may even give a clue of an approach of what they can do next, so maybe even to go beyond that.
Brian?
So one of the things that wasn't specified in the plan was what genomic platform is used. And it leads to the question, is great the enemy of good? So should this be something that is introduced stepwise, in which one initially trials structural changes and then builds on that with RNA and more sophisticated sequencing techniques to get it started, to show proof of concept? That may be something that might be more palatable than starting with whole genome sequencing right off the bat.
Yeah, so just a comment. I mean, obviously, what I put out there is sort of agnostic with respect to what testing is done. And it's really, my thinking is that that'll be driven by both the science and the marketplace, right? So doctors will obtain or order what tests they believe are clinically useful in managing their patients. And that, you know, over time will involve a variety of different kinds of test platforms. And the issue is, you know, to be able to capture what test is being done, what decision is being made based on that test, and what are the outcomes to the patients, you know, who have an action taken based on that interpretation.
So what do you predict, which platforms are going to be available in the next couple of years? I think that, you know, being able to replicate what Michigan has very, very skillfully done on a national basis for clinicians, you know, in the community is probably not going to be easy. It's probably going to be easier for them to deal with commercial entities that are...
Absolutely. So, you know, commercial entities will go where they think they can make money with whatever platform they think is going to be clinically useful, which at the moment, you know, is mostly mutation profiling.
Exactly. Skip?
Yeah, I think Brian touched on a point that's really important. Carl Morrison mentioned earlier today, what is the test? What test are you talking about? So, you know, we've neatly jumped past that, or we're agnostic to it. But I think Rich and I were talking in the lunch line. Perhaps I think we should be careful about arguing that we should do something because what we've been doing hasn't worked. But the fact of the matter is we have an opportunity to take advantage of the vast majority of patients who are getting treated for cancer, getting diagnosed with cancer, and about whose course we're learning very little. And so that by itself, I think, is a reason that energizes me about this idea. You know, there are lots of details, but there are a lot of smart people that can work on those details. I think this is a sort of paradigm-shifting approach that has a lot of merit.
You know, one other comment I'll make is, you know, one of the issues that also came up over lunch, you know, that distinguishes this country from many other countries in the world, of course, is that pathology in this country, like many other areas of medicine, is kind of the Wild West. I mean, it is up to, you know, individual laboratories to introduce whatever tests they think they can come up with that they can make a claim for and get those into the marketplace, whether or not it's proven that they're useful for anything. Now, interestingly, if you look at the way they've introduced genomic testing in France, and I mentioned this in my talk at Asco as well. So, I mean, the French National Cancer Institute has set up 28 regional genomic testing centers for the country. They all do the same test on the same platform, so that any patient in France who has a tumor for which molecular profiling is known to be beneficial will get a reliable test result from a laboratory that's using harmonized SOPs. I couldn't imagine that happening in this country. And so the limitation that we have, one of the limitations we have, is that there are very few standards that have been established for how tissue should be handled, what is the appropriate molecular workup of any given tumor diagnosis, that is, what tests should be done, with what platform, how it should be reported, what is the actual definition of a positive test or a negative test. And I think there are opportunities for some of the pathology professional organizations like CAP, like the Association of American Pathologists, to work with the clinical oncology organizations to develop guidelines and proficiency testing standards that laboratories would be asked to follow in order to be declared appropriate testing facilities for this kind of molecular workup, and their reimbursement could be linked to meeting proficiency standards. And that would help improve the quality a lot.
So I see a challenge and a risk with a national coordination or nationally coordinated approach. One relates to this issue of how good is the tester, and then how good is the testing, because even if everything is certified and you replicate it in 12 different laboratories, there's a bell-shaped distribution and there's going to be outliers on both sides. Sometimes those are random. Sometimes when you come back 6 months, it's the same sites, which are the outliers. So this could be anonymized to the point that it only establishes for us and for CMS that the overall outcome is better or worse than a 9 or 12% response rate seen on phase 1 trials, so that we knew we're doing at least as good as phase 1 trials. Or it could be used to improve the whole process, which means that we have to be able to mine data for centers, testing sites, physician decision-making groups that are off target in terms of the norms of the distribution. So to me, it seems to me it's the opportunity, as Roy mentioned earlier about immunohistochemistry, where it was a boondoggle for years until we really understood how complex that was. One possibility here is we shorten that cycle and improve dramatically efficiencies. And one other topic I think is a risk is that if you're the commercial site that's seen as the outlier, if you're a pharmaceutical that's deposited your drug and it's been used for X disease, and now you show that and as I'm sure that will happen, it actually has adverse consequences for individuals with certain mutations. You're taking a risk by participating.
No question. But, again, this comes back a little bit to Brian's point about value. There's a tremendous amount of medical resources that are consumed in this country practicing medicine is completely non-evidence-based. Interestingly, one of my colleagues at U of C, Rena Conti, who's an economist, published a paper recently in JCO about off-label use of anti-cancer drugs. And then there was another study just presented at Asco last week about off-label use of anti-cancer drugs. And they both show about the same thing, which is roughly 70% of cancer drug use is on-label. 30% of it is off-label. And of the off-label use, half of that is not supported by any evidence, by anything in compendia, anything in the literature. It costs about $8 billion a year to reimburse for this 15% of cancer drug use that is non-evidence-based. And we can't afford that. I mean, we have to find a way to improve the value equation. And one way of doing that is to have prospective registries that record outcomes that can actually then allow informed reimbursement decisions to be made. I mean, sure, the drug companies are taking some risk, but they're also potentially benefiting from, observations that may lead them to, an off-label use that they could develop into a new indication, and, that kind of thing.
So I think that's a great point. And actually, the whole issue of standardization of clinical care is very, very important because increasingly we're going to be mandated to develop care paths and other algorithms to standardize what we do. And I think that one way to potentially validate such a national network is to incorporate it into a care path of standard, this is how we think we should approach oncology care going forward. But the point is that doing everything as a one-off in the absence of any sort of standardized algorithms will not get us to where we need to be.
So if we get through this process, somebody above our grades is going to ask, well, what are the endpoints, and how do we know we've succeeded in the process? So is it just a rational use of resources, so we know better how to use tests and better how to use drugs? Is there some way for CMS or FDA to help us by validating or approving something? Do we want an approval process, or do we just want a mechanism to help the nation learn about this new technology? Don't all speak at once, but.
Well, I mean, I'll start, and we can go down the line. I mean, so, of course, you've asked a critically important question. To some extent, I think we'll know it when we see it, because hopefully we'll have a tremendous amount of additional information compared to what we have easily available to us now. I think to some extent that if we went this way, it'll be more useful to inform reimbursement decisions than it will to inform regulatory decisions on drug and test approvals. Because, it won't take a huge amount of information to make the observation that, we gave this drug based on this test to 1,000 patients and nobody responded. And therefore, we probably should quit doing that. And I think that, the payers will pretty quickly catch on to that. What it's going to take to inform a regulatory decision, I think we heard from our FDA colleagues this morning, and it's going to be a little bit of, it when you see it, in terms of how well characterized the patient population is and how robust the evidence of anti-tumor activity of the drug is. I think at the very least, if we had this type of registry thing, it would enable us to develop more informed hypotheses of what studies to do, so that we can do studies that potentially have the highest payoff down the road. But it is at least conceivable to me, that if you saw a surprisingly robust and reproducible level of anti-tumor activity in a rare subpopulation defined by a molecular marker treated with a drug outside of a clinical trial. How many times did you need to get a CR from MOP chemotherapy to know that it was a good treatment for Hodgkin's disease? And how many episodes of testis cancer needed to be cured with platinum-based therapy to know that you were looking at something that was new and different? And I think if we, would be able to demonstrate that for, well-matched drugs to the appropriate target in well-characterized patients, it might be persuasive enough for a regulatory decision.
I think, as Rich said, I think that rather than being persuasive for the regulatory decision, which could happen, I think, on rare occasions, that it would be of more value to design the clinical trials, particularly phase 3 clinical trials, because these trials are so expensive and the companies have so much invested. And if you actually look at the number, and people like Rich would know better than I do, that there has been more surprising number of negative phase 3 trials, which you would think that there would have been enough signal from the phase 2 studies that there should be very few negative phase 3 trials, at least if the ideas you think are really that good. If you had this national registry, it may really add a lot of information to the phase 3 trial, and then the company themselves could get the information. Now, the difficulty may be finding enough patients for a phase 3 trial that are in relatively smaller subgroups, and that would have to mean that the larger cooperative groups will have to be more nimble at doing this kind of analysis so they can group their patients in such a way to make heads or tails of it.
Skip?
I agree with both Max and Rich, particularly about the positive aspects of leading us to both hypothesis generation as well as perhaps defining a drug that would be useful based on 1,000 N of 1 experiences. I think the experience of the 2 reports that Rich alluded to on how we use drugs without any rationale, we should be sobered by that. And I'm not as confident that Rich might have implied, that if we have 1,000 patients on this registry trial with mutations in gene X who are treated with drug Y who don't respond, that the payers will have the organization or the gumption or the whatever to say they're not going to keep reimbursing it. Because if 15% of our chemotherapy is now reimbursed based on no evidence at all, that's scary. And that's a problem that we also need to undertake.
It's after lunch, so I'm sure that blood is just starting to get back to your brain, but I'm delighted to take questions if I can get some hands raised in the audience. So we got 2 here.
I think our discussions about payment from and value versus volume will be solved by CMS itself. In fact, I think CMS is moving towards capitated payments. So as a result of this whole problem of paying for drugs that are off formulary or paying for drugs that are not approved for something will simply be shifted back into our lap. And we will have gigantic health systems with capitated payments, and we have to manage the resources ourselves, and that's how CMS will fix this.
Carl?
As a pathologist who's a part of that wild, wild west, I would like to tell you that I really like the idea of the National Registry or Formulary of Drugs and Mutations, because that defines the test, okay? And the only thing I think you don't want to leave out of this equation is, if you have defined the test, make sure that you put a system into place so that whoever's doing the testing is doing the testing of the highest quality, because you now have defined the test.
Yeah, no, I mean, I couldn't agree more, Carl. I mean, I think this is something, the clinical oncology community can demand this, but only the pathology community can actually make it happen. And I think this is something that, you and your colleagues need to really be talking about in terms of, ensuring that the testing that is provided by pathology laboratories is not only analytically of the highest quality, but actually has some evidence, to establish a claim for its clinical necessity.
So I think, too, that, pathology is always somewhat disconnected from the clinical evidence. And we're always looking for what is the test we should be doing. So in this program that you all have outlined, at least you've defined the test, and from there we can make the process work efficiently and accurately.
Neil?
Just one comment about incentives and sort of carrots for making a system like this happen. With regard to the payers, and I don't think this appeared on your list, a potential incentive is that not only that they will get information that will be useful in making future decisions, but it will enable them to say no to people, to requests that are not part of this system. And the system will be characterized by credentialed investigators who we can believe their data, and probably only specific clinical contexts. So there is this other, huge motivational factor, I would think, in that it will allow, them to sort of the payers to fend off these Wild West requests that are deluging them, every day now.
Pat?
The one thing you have to make sure if you ever move forward in this is to be able to have best-in-class of agents and be able to pull them out and bring in better ones as a new best-in-class or a better best-in-class becomes available. And then you always, again, have to worry about drug combinations because so many of these patients have multiple actionable mutations and how you're going to move forward with that as well.
Pat, who defines the best in class, and how would that be done?
Wow, that's a challenge, I guess. Jim, you could help me with this a little, but obviously that's a moving target. I think it's not only, a lot of it is based on preclinical data, but also preliminary phase 1 clinical data as well to confirm that based on what you identified preclinically, you could confirm clinically in terms of target effects, as long as you're doing the right phase 1 trials, in terms of exposure, in terms of acceptable toxicity, many factors. I mean, Jim, do you want to weigh into that at all? No. Or yes, here. I mean, it's hard.
So I would say, I would emphasize that what Pat said about the using the phase 1 data, give it within an example, and then I'll say something about the preclinical. So we're now it's at least the third generation of HSP90 inhibitors, maybe fourth, but third certainly. And I know about this because the NCI invested a lot in the first and the version 1.5, right, to know definite benefit, right, to anybody. And to the point where we're almost ready to just, we're not interested in HSP90s inhibitors anymore. And yet now there is at least one drug that is remarkably effective, right? Same target. Why? It's clearly the best in class, right? And that has taken a decade of clinical trials. I don't think it was because of preclinical investigations that occurred, but so I think you have to do and be able to switch out drugs in large trials to be able to find ones as the data becomes known. The second piece is that we've engaged, I think, relatively effectively. I'd like to do more and try to do more open preclinical stuff, comparative, I call it comparative pharmacology, right? So we were on the verge of spending $5 million to support NSABP to do an early, it wasn't an adjuvant trial, I think it was a neoadjuvant trial of GemCis or GemCarbo plus iniparib, right, the BSI drug. And then we did the data, we developed an assay, and it didn't hit its target and compared to all the other compounds that were available. Stopped the trial, saved the government and anybody else lots of money. Of course, it cost Sanofi $500 million because they were wrong to take the drug on. We've just finished another series of studies presented at Asco of all the available MET inhibitors. And unfortunately, there's one of those agents that, in fact, CTEP took in, and trials are ongoing. It does not inhibit phospho-MET at all, in vitro or in vivo, right? And so the problem is that the companies all know this information, but it never becomes public. So one of our tasks, NCI's tasks, is to try where it's appropriate to do the studies preclinically and make it all widely available, so investigators in their laboratories or when it comes to the clinic know what to avoid and what agents are most useful. But I think that in this context, Pat is right, that you just have to see the early, it may take 3, 4, 5 years, but over the course of, say, 3 to 5 years, you know which of the phase 1 agents are better than others.
And you're probably going to have several same of class, because like right with our Stand Up to Cancer MRA project right now, we realize that not all MEK inhibitors are created equal, not because of potency or whatever, but there's certain subclasses of actionable mutations that you would think MEK, but one MEK does it and another MEK doesn't. And so, I mean, all of this is a science, and it's not, I applaud you for this concept, but you have to make sure you're constantly thinking the variables, because the last thing you want to do is consider it a negative study when you haven't looked at all the,
So one of the advantages of the concept is that you're taking the decision-making away from the individual clinician. and you're putting it into kind of a national tumor board analogous to the
Hopefully,
local tumor
yes.
board that Michigan's done. So you have a
Yes.
validation of process, I think, which is a pretty big deal, which hasn't been discussed very much today, that taking the decision away from the individual clinician will hopefully reduce that
Hopefully.
15% and $8 billion of illogical use of therapeutic agents.
And hopefully you're going to figure out all these things because you're going to have the tissue and you're going to have all the data
Right.
under one roof.
But you see, the other aspect of this is, quite frankly, I don't know that it will be possible for us, or that we will even need to figure out all these things in advance of these drugs making their way into the marketplace. But once they're in the marketplace, sort of the big data approach will really facilitate figuring this out. I mean, if you had, if there were 3 MEK inhibitors, available, commercially available, and you had data on, 50,000 patients who got those MEK inhibitors, and you could look at the data and see who got which one based on which test and what were their outcomes, you'd probably be able to see pretty quickly a trend as to which one is going to work best in which circumstance. And that may, ultimately result in decisions being made going forward that, could not have been made until those analyses were completed.
This was a terrific discussion. We're going to move on because it's 2 o'clock, and I'm going to invite the last panel of the day with moderator Lindsay Harris to come up and talk about clinical trials and testing.
Appreciate your sticking it out to the end. And while all of our panel members are assembling, actually, I think I'll be standing because there aren't enough chairs. What we're going to do as we wrap up is to really discuss some of the pragmatic issues around this entire forum, specifically how do patients, physicians, and the community embrace genomic testing and considering using the information for making clinical decisions. I'm going to go through and have each panel member introduce themselves, and we do have some specific questions. But I also want to just give you a heads up that we're going to have a patient and his physician from the local area give us an anecdote or a story that I think will help to put it into perspective for many of us who I think are struggling with this whole situation. So now that everyone's here assembled, and do we have Paul and there they are. Great, good. So you can make your way up to the podium, unless maybe you'll just sit. Maybe that's better if you just sit there, and then we'll have Mary or one of our. Great, Mary. Can you bring them the microphone? Excellent. So first of all, I'll just have all of the panel members introduce themselves. So I'm Lindsay Harris. I'm a medical oncologist. I direct the breast program at the Seidman Cancer Center and co-lead the developing NIH breast program here at Case Western. My background is as a medical oncologist and a translational researcher. I've done a lot of work in biomarkers over the years in genomics and have worked a lot in the cooperative groups. Dr. Shilsky and I have worked together for many years, and I think what's interesting to me is that in many ways we're just talking about another biomarker, except we're talking about a very complicated biomarker. And so for many years I think we've struggled with biomarkers being essentially peripheral to the practice of medicine, to now where biomarkers are actually becoming very integral to the practice of medicine. And to the extent that at least a decade or so ago, Bob Bast and Dan Hayes sent up one of the first panels called Asco Tumor Marker Guidelines, which was really to help practicing oncologists try and understand how to use biomarkers in the setting of clinical care. So not so much addressing specific issues around how the biomarkers were tested, all that was assumed, but really how do we use these sorts of molecular tests, immunohistochemical tests, et cetera, to make clinical decisions. And some of the more visionary people, such as Dan and Bob Soon-Paik and others, have published guidelines for levels of evidence for biomarkers that are, similar to what we think of levels of evidence for clinical care. And now we're faced with, I think, a tsunami of information on biomarkers. And again, the cart, as it does get ahead of the horse from time to time, I think we really need to consider all we've learned about how to use molecular testing in the setting of clinical practice. So I'm hoping that throughout our discussion, we'll be thinking about some of the levels of evidence and the rigor with which we've tried to evaluate tumor markers going forward, and then we can talk about some specific situations during the panel discussion. So Candace, would you like to go ahead and introduce yourself?
Candace Johnson. I'm the Deputy Director at Roswell Park in Buffalo. Together with Carl Morrison, Skip Trump, and the rest of our Roswell Park pals, we're trying to develop personalized medicine not only to bring to our patients, but also as a research initiative. I'm a pharmacologist, and in GU cancer, I've most recently been interested in genetic signatures that might guide therapies in bladder.
My name is Gerald Beppler. I'm the president and CEO of the Karmanos Cancer Institute in Detroit, Michigan. My area of interest is lung cancer, and I have been working for almost 2 decades on trying to develop predictive markers for therapeutic decisions in non-small cell lung cancer.
Hi, I'm Michelle Mcgowan. I'm an assistant professor of bioethics here at Case Western Reserve University. I'm the site PI for a National Human Genome Research Institute study called Anticipating Personalized Genomic Medicine, where over the past 3 years we've been conducting interviews with a variety of stakeholders who are interested in seeing the translation of genomic research into genomic medicine across a variety of specialties. So I'll be speaking to some of the ethical and social implications that they've raised in the context of these interviews.
Hi, I'm James Lee from University of Pittsburgh. I'm a medical oncologist focusing on phase 1 and colorectal cancer. Regarding the genomics, I recently opened a cancer genome registry protocol in Pittsburgh. I'll be approved. So I think more relevant, all the discussions today, I think gives me a lot of practical points.
I'm Pat LaRusso. I'm a medical oncologist at Dr. Beppler's Institute, Karmanos Cancer Institute. I run the phase 1 clinical trials program there, and for obvious reasons, because of drug development, we're very interested in genomic signatures and drug response and drug toxicity.
Thank you, everyone. And so I'll ask Paul and Judah if you want to just give us a few minutes of your time and describe your situation. Judah, maybe you would give an introduction about Paul's case.
I'm Judah Friedman. I'm a medical oncologist with University Hospitals. I work out on the east side of Cleveland. And I met Paul. This is Paul Gothard. He's a professor of music at Lake Erie College. And I met Paul just over a year ago when he was being evaluated for difficulty with breathing. He was having coughing and was being diagnosed initially with pneumonia. Further testing revealed evidence for a type of lung cancer, non-small cell lung cancer. And after extensive evaluation and review, we felt that there was a possibility that the cancer was confined to the bottom of the left lung. And however, at that time, he was having quite extreme difficulty with breathing, producing copious amounts of fluid with coughing, unable to sleep at night from this problem. And Paul described some of that in more detail. But basically, we decided to give him chemotherapy initially, which caused a very rapid improvement in his symptoms, and he was able to move forward with surgery. And ultimately, here at Case Western, had to have required a pneumonectomy, removal of the entire lung due to involvement of the tumor in the left lung. Afterwards, he recovered and did quite well, and we continued with some additional chemotherapy. So I wanted to maybe turn it over to Paul to describe some of his initial treatment and how he was doing at that time.
Thank you, Dr. Friedman. Yes, my name is Paul Gothard, 61 years old, college professor most of my life, and if not all of it, and never smoked. But indeed, the surgery went very well, and we thought things were going just fine. It continued on post-surgical chemo, and as a result of a CAT scan, we found spots on the lower right lung. And so we proceeded to, with other treatments, thinking that it might be an infection, injury from surgery, and lo and behold, a bronchoscopy biopsy revealed that it was indeed cancer. So we went to, with the bioptic material was analyzed, and Dr. Friedman informed me that I was a candidate. I had the HER2 receptor, and we went on Herceptin alone. It wasn't coupled with standard chemotherapy. My main problem was more the fluid, which was preventing any oxygen carbon dioxide exchange. And the Herceptin within 4 days by itself started to eliminate the fluid. I had already begun a semester of teaching at the college and would have very much liked to have finished the semester. I had a number of pieces in composition that I wanted to finish and premiere. And I responded very well to Herceptin. Unfortunately, the cancer is all through the entire right lung, so the Herceptin is only holding off the worst of the symptoms. So I just wanted to be here because I am, as I mentioned to Dr. Gerson, probably a textbook example of the textbook that hasn't been written yet, both in terms of the fact that the targeted Herceptin by itself eliminated the fluid, and I've gotten 4 more months of life because in February I wasn't able to roll over in bed without going into complete oxygen deficit. So I was able to finish out the semester. I was able to conduct the work. We're at this point, the Herceptin might be losing some of its efficacy in as much as some of the fluid is coming back, but the quality of life has been exceptional. I've been fortunate enough not to have any side effects from it. I didn't have any from the carboplatin and Alimta that I was originally on, so that made it all very, very good for me. And so I guess I'm here. I am here as a result of the Herceptin treatment, and I'm here also to say that it was a problem, finding coverage. To cover the Herceptin. I'm on hospice, and hospice has stepped in, and they are covering the Herceptin treatment for as long as it remains working for me, fortunately, because I was turned down by all other insurance agents. So that's it. I mean, I'm very grateful. If I can be of any influence on this, here I am. I'm still alive. I'm still breathing, rather shallowly, but I'm here, and encouraging this wonderful movement that's going on here, because it certainly has made a difference in my life.
Well, thank you so much, Paul and Judah, for sharing that. And I think this points out a couple of really important messages here, which is you had a mutation or an amplification, I guess, that was not expected based on your type of cancer, and that it was found by an oncologist who was thinking about genomics and what other targets could there be. And you responded immediately and dramatically to that targeted therapy, and unfortunately your insurance company refused to pay, and your appeals process was refused through the pharmaceutical company as well, and it wasn't until Case Western Reserve Hospice decided that they would pick it up as palliative care that it was actually supported. So I think these are some of the questions that in the situations that we're grappling with, and I think Dr. Shilsky's, very visionary approach to really trying to make these sorts of therapies available to patients with specific targeted lesions, I think is really, I'm optimistic that those sorts of approaches will, allow your opportunities to be greater in the future, and folks like you. And so I did want to just go into some of the specific questions. And again, this is more of the pragmatic session. So we're going to ask individuals who have started off the conversation about certain areas, the Genomics Tumor Board, the ethical implications of testing for patients, what role some of the labs and reference labs and biotech companies should be thinking about playing. What are the clinical trial designs that we want to be thinking about going forward, and now that we've, really talked about some of the bigger issues. And then, from my perspective as head of the Tumor Marker Guidelines Panel for Breast Cancer, developing the new set of guidelines going forward, how do we really think about educating the population of oncologists and providers in the country as to how to think about using these tests in clinical practice. So these are our outlines of questions, and as I bring them up, I'll point to an individual, but really all of the panel members will be asked to address the question if they'd like to. So I guess the first question is toward Dr. Lee. How do you think these genomic tests should be made available to practitioners in clinical practice, and what sort of approach have you taken at your institution?
I think this is quite recent happening. Last year, November, Pittsburgh Molecular Pathology, they set up a CLIA certified next-gen sequencing capability, but not like whole genome sequencing. We try to approach more the high-value target genes. So the first version was 46 genes mutation profiling. And initially we started kind of the most common, like three, like a colorectal, lung, or breast cancer sequencing, but right now expand to all different types of cancer types. But I'm a medical oncologist. From my point, I'm ordering, then I will get 2 weeks later the sequencing mutation profile. And also phase 1, we are looking for what will be the better phase 1 approach or clinical trials. And what, through the discussion, what we got was that we need to build up some type of some instrument. We collect all the sequencing data, matching with the clinical data, so that we can, such as we have a several 100,000 patient database accumulate, then we can query on time how many patients with a certain target mutation, so that we can have a very the active the database of a certain trial or even treatment approach, like Asco is planning to do. So that was the cancer genome registry I just briefly mentioned. But we are limiting to cancer cell sequencing, not the whole the germline. That's the way we could sail through the IRB approval easily, rather than kind of very difficult consenting procedure. So this will be the patient consent and give us a full access to clinical and genomic, cancer genome sequencing. And the next step was that, so I'm a medical oncologist, not the pathologist. So when I got the report, the first question is that what should I do with this data, and what is the implication? How does it translate into actually my decision for my patient care? So what he did was that, I could not solve by myself. So we kind of invite all different parties, and did actually launch the first genome tumor board like a couple weeks ago. It's the first I presented my patient case, colorectal patient, and we actually initially invite from even the preclinical folks, because my patient carried a very unique mutation. I could not find any real clinical trials. So try to get some ideas what's any most current preclinical data about this mutation. So we had a quite productive discussion, but still I think it left us more question than answer. So what we should do. So I view this as a more likely good starting point rather than something we can really actionable yet.
So Pat, do you have I wondered if you have an approach, given that you have a very active phase 1 program at Carmanas.
We just started implementing a more aggressive, prospective way of doing this. So if it's clinical trial selective or specific, based on a couple trials that we have, for instance, our BRAF wild-type trial in melanoma, we're doing extensive sequencing, whole genome exome RNA-seq, very hypermutated tumor. We're working in conjunction with TGen and Phoenix that I think are probably as good as a rule, and maybe a little better. The verdict's out. So anyway, but I won't hold it against him. But with some of our other tumors, when some of the other trials that we're actively developing, depending on the tumor type, we're doing limited profiling, capturing tumor on those patients so that we can do more extensive profiling based on the results. And for instance, we just got approval from Jim's group at the NCI to look at bladder cancer. And we're doing extensive biopsies on those patients, but we're going to initially profile just for TSC1/2, predicated on those, and then everybody will give us extensive tumor, and then we'll look at responders versus non-responders. We're going to be hopefully doing a similar thing with a MEK-AKT triple-negative breast cancer trial. For all the other patients right now, we've just entered into a relationship with one of the large companies, and we're going to be doing 3 different types of platforms so that we can do a prospective comparative analysis as we move forward, looking at the differences in those different platforms. And hopefully at the same time, because we're very, very blessed in that we have a large cadre of trials right now, be able to use some of that data to inform us as to which patients should go on which trial. But I'm not convinced that we know what the right platform is. So we're going to be doing 3 separate platforms and hopefully be able to come up with some prospective, meaningful data, because nobody's really looked at prospective platform analysis. Yeah.
So I'm going to open this to the other panel members and also to the audience. I think the pathologists have great comments to make, and so I'd love to hear any questions there or additional comments. Candace?
One of the things, I think one of the issues with this information is what do you do with the information once you get it back from the genomic lab? What does the pathologist do with it, and what does the physician do with it? We're trying to develop perhaps an omics solution, a bioinformatics solution to help this, to help the physician be able to look at the literature associated with the particular mutation involved, to help the pathologist understand it a little bit better and what clinical trials are available around the country. Because I think that none of us know what all these things mean in our specific discipline. And so I think that, and we can all work together on this. We're partnering with My Cancer Genome and Vanderbilt that across a few of tumor types has really done a phenomenal job, but there's still more work to be done.
Oh, yeah, and I mean, Arul can tell you, and anybody that does tumor boards, Friday I was on 3 hours of tumor boards. Each patient takes at least an hour, and, because we don't know enough yet as to what to do, and, when you have Neil Rosen and Dave Salad on your tumor boards, they could last way longer than an hour. Yes. But there's a limit. Thank God Jeff can cut them off. But, there's, the verdict's out. You don't even, it's very challenging. Yes.
Any other comment? Oh, I'm sorry, Jerry.
Yeah, I just wanted to tell you a sort of personal experience, because I think we can't underestimate the importance of biospecimen collection and preservation, which we in a sense have glossed over to a degree. Yes. I just want to tell you that so we did an analysis of 2 biomarkers from lung cancer specimens collected across the world. This was an analysis of 1,000 lung cancer specimens that were surgically resected coming from South America, North America, and Europe mostly. And we analyzed 2 biomarkers by an automated method. So meaning at least the methodology should be solid. And then we gave these data to a biostatistician, and the biostatistician sorted these specimens exactly into where they came from which institution, without knowing it. So meaning really the way biospecimens are processed has an enormous impact on the level of gene expression. I can't speak to the same effect from a point of view of mutations, but I would expect if you go for RNA, you would find the same result. For DNA, which is more solid, more stable as a molecule, you probably won't find this. But a lot of the molecules that we are actually talking about that we want to be using for therapeutic decision making, ultimately we have to really measure them at the level where they are, where they have action, and action is at the protein level, and actually is in the context of complex protein complexes and function. So the ones that we are analyzing right now and that we have great success with, like vimorafenib with BRAF mutated melanoma and EGFR mutated lung cancer, is just sort of the tip of the iceberg. These are the ones where the mutation that you see is directly responsible for a very, very strong and impactful biologic function where we can do this. But I think once we get to multiple genes at multiple levels, different levels of expression and mutation, this becomes increasingly complex, and we really have to be careful that we design clinical trials in such a way that we can really answer the questions we want to answer. Just to get back to the Asco meeting, which was talked about a lot, the last Asco meeting presented trials from lung cancer using gene expression to make therapeutic decisions from Italy, Spain, and the United States, and all these three were negative. And these were based on 15 years worth of biological data and information. These were prospectively designed trials. All three of them were negative. So I can't, yes, we are very enthusiastic about where the future will be with genomics, and believe me, I'm extremely enthusiastic about this, but we have to be very careful when we try to translate results like crizotinib and ALM rearrangements and EGFR and Tarceva into other diseases. Just be careful.
And I think one of the other concerns is using proteomics prospectively, especially because of tissue handling.
So I think you really pointed out, the last 15 years of work with biomarkers is, just discovering that pre-analytical variables and tissue handling and testing is just as important as, the drug that you're, and all the complexity of the drugs that you choose to target that lesion. And we've learned a lot, and I think, the ASCO CAP guidelines are an example of how, ASCO and College American pathologists have worked together to try and come up with a unified approach to, and we're just talking about single genes. We're talking HER2, we're talking ER, we're talking, in colon cancer, certain targeted mutations. So I think, these are the sorts of things that we need to envision going forward, where our educational bodies, regulatory bodies are really going to be scrutinizing carefully both sides of the equation, the analytic side of the equation and the therapeutic side of the equation, and how we really bring those together optimally to, in the setting of, the registries that Rich is talking about, and even the clinical trial initiatives all require this sort of analytical and, careful perspective evaluation. There's no free lunch, unfortunately, although you've had it. So anyway, now we're going to switch, unless there's other comments from the audience, I think we'll switch topics a little bit. Again, this is an area that was touched on a little bit in I think the third panel, but I would really like to have the opportunity to have Michelle Mcgowan speak to some of the ethical issues surrounding genomic sequencing, and we've had the pleasure here of working with Patty Marshall, and Michelle from the Department of Bioethics, who've been working on, looking at the implications of genetic and genomic testing in international settings with Francis Collins, and are sitting on national and international panels to discuss how, we move forward with these sorts of endeavors. So I think this might help you, Rich, too, for what you were thinking about.
Great, thank you. So there are a variety of ethical issues that I could touch on, so I'll touch on a few of them and then focus in on the one that I think is probably the most pressing. So first, we can't talk about bioethics without informed consent, which we heard already earlier today, that there's a lot of debate about whether informed consent is necessary for quality improvement studies versus clinical trials and involvement in clinical research. And I think that I would encourage this group to look to the bioethics literature on biobanking with genetic information. There's a really rich history there. In terms of demonstrating that there's variability across sites, which we heard even on the panel earlier today, in terms of whether there's a need to institute informed consent on a study-specific level versus broad consent when you donate a sample to a biobank, for instance. And I think that in the bioethics literature, what we've learned is that participants in biobanks really do want to be part of the process. They would rather be asked, even if they might have consented anyway. So even if there is going to be de-identified data available for research purposes only, in this era, I spoke with a genetic researcher last week who says it's a fallacy to believe that we could ever keep this information de-identified. So even if you as a research community understand the difference between germline genetic information and somatic information, that might not necessarily be a pertinent distinction that's well understood by the patient populations and the research populations that you're interested in enrolling in studies using genomic information. So this is something that will need to be better developed in the context of the informed consent process specific to cancer genomics, I think. And we also heard earlier today about the questions around health economics. I think I'll just touch very briefly on that, that in the literature, there's even a citation in your pamphlet today around whether health inequalities could arise in the context of cancer genomics, and that's written from the perspective of European and Canadian researchers who are working in national health services. And so the questions around health rationing and health economics take on a different there's a different tenor to the discussions there than we might have here in the US context, where the potential for health inequalities is actually much larger, I think, in cancer genomics. My personal concern in this area is that access to participation in genomic research, to clinical trials, as we already heard today, to genomic testing, and to genomic information is going to be very, very circumscribed in the sense that most patients are not going to necessarily have access to this information, and a lot of clinicians working in community settings won't either for quite some time. So to lessen the risk of health disparities being exacerbated in the context of cancer genomics, I think we have to be mindful about the impact of the uneven distribution of healthcare resources and research resources in cancer genomics in particular. Some of the scientists that I've interviewed in the context of my own study have argued that there's potential for some cancers to turn into orphan diseases because of rare genotypes that can be identified. And there also may be an exacerbation or a creation of new underserved populations of cancer patients. So in an effort to recognize the justice implications of cancer genomics as it moves forward, we should keep these things in mind. I think that I'll end by arguing that I think the main concern that we've seen, if we think about the popular media coverage of personalized medicine and whole genome sequencing, has been that personalized medicine and precision medicine are available to those who can afford it. So if reimbursement remains a problem, then there is an inevitable access issue that will arise in where people who have $9,000 available for sequencing of the whole genome will be able to have access to better care and better information than those who don't. So I'll end on that point.
Great. Thank you, Michelle. That was really spectacular review of some of the ethical issues and access issues. So are there any questions from the audience about or thoughts about concerns about ethical implications? And we may also ask our panel members who talked about offering the sequencing what they're doing to really address some of the ethical implications. So one of the things that I'll just bring up while people are thinking about that is, so we have a proposal that we've been moving forward where we have actually a genomics navigator whose role is not only to consent the patient, but also to do interviews with Michelle and her team about, really what are the implications of these information to patients and providers, and how do they want the information delivered. What, as Arul said, there are different levels of meaning of these lesions, so things that we know what to do with, things that we know about but don't know what to do with, and et cetera, et cetera. But there's also going to be things that are discovered that may have relevance for that patient's risk of heart disease, for example, and what is happening with all of the genetic information that comes from whole genome sequencing, et cetera. How is that being stored? What's the access issue? Does the patient want to know information? It's very similar to the discussions we've had about the BRCA testing over the years, so I think this is really sort of expands that even more dramatically. And so, I don't know, Arul, maybe you could take on that question about, what kind of discussions you have with the patients who are undergoing sequencing about potential, incidental findings or germline findings. Does someone have a microphone?
So that was certainly an issue with our IRB when we initiated this protocol, the fact that we were sequencing the germline and comparing it against somatic alterations. So that is one of the reasons why our protocol has built in a meeting with the genetic counselor before we enroll patients. So that's certainly one aspect to that. So our bioethics team, which is led by Scott Roberts and Scott Kim at our institution, they have developed a nice, what they call a flexible consent model, whereby the patient has the choice of merely consenting to cancer-related findings, things that are related in their somatic genome as well as in their germline genome that might be related to their cancer per se. They can essentially opt out of having things that are related to their family or hereditary issues in general, or things that might not even be related to their cancer, like heart disease, Alzheimer's, and so forth. So they have the ability to opt out of those particular findings if we have that. So they sort of, I think that the thought was that they would be empowered to choose what type of information that they'd like to have. But the minimum is that they would be conveyed information that was cancer-related, would certainly be conveyed.
Thank you. And are those being stored in silico, or do you actually toss that data?
We store the data. And we've told them that we would store the data, as well as that we would deposit the data upon publication to restricted warehouses like dbGaP and so forth, but not necessarily make them completely publicly available.
Gerald?
We've been talking about this always in the context of this is better, and are we withholding information, or do we not give access to patients who don't have the financial means to actually do this? I just wanted to point out that, again, at Asco this year was the first time a trial reported, to my knowledge, where actually genomic testing was done to select patients for therapy, where the outcome for patients that were tailored, where the treatment was tailored by the genomic information, actually resulted in worse outcome. So they had less progression-free survival and actually, in fact, less overall survival. So it's not necessarily always goes in one direction. It can go the other direction, too.
Thank you. Did you have a comment?
This is a follow-up question on the choice you're giving patients to either receive results specific only to their somatic mutations or their germline. I'm just curious as to whether or not you're screening for germline mutations or how you're defining an incidental finding in those circumstances.
Yeah, I think that is certainly a moving target in terms of what we're defining and how we're analyzing the data. And there have been some consensus reports on what types of germline findings should be reported in general. So that's really one of the reasons why we're involving a team of genetic counselors to help us make those potential decisions as to what findings should be potentially reported to an individual patient if they opt to having that information. So they eventually make that decision in conjunction with their treating oncologist.
I guess I'm wondering though, are you screening up front for a specific set of germline mutations?
No, because our assessment is unbiased in terms of the genome germline assessment as well as the somatic assessment. So, for example, we would see a BRCA mutation through the analysis, basically.
I just wanted to add that for those of you who are familiar with thinking about what information should be returned, what incidental findings might be considered worthy of returning to a patient. The American College of Medical Genetics recently released a set of guidelines around what laboratories should return incidental findings, and they mainly pertain to cancer-related findings. But there has been a real upheaval in the ethics community and genetics community around this recommendation by the American College of Medical Genetics, whereby some laboratories are actually adding in an opt-out clause to their own informed consent documents because they don't want to follow the recommendations that were given by the college. So this is a problem that is still a moving target across specialties. In both the research and clinical environment. There's a large consortium out of the National Human Genome Research Institute's ELSI program, the Ethical, Legal, Social Implications program, on return of results that will have a number of publications coming out in the next 6 months to a year. I think the first couple have already just came out last week. So keep an eye out for those if you would like to follow the debate further.
John?
I'd be interested in the perspective of any of the panelists as we're talking about the development of national genomic networks and repositories of molecularly targeted therapies. Are there unique challenges that you see or perhaps special considerations that have to be addressed to ensure continued effective inclusion of children in the process, and so that our pediatric patients are in real time also benefiting from this process that's being established? And I know there's no right answer, but I just wanted to ask.
It's a great
Arul,
question.
You've been doing some pediatric cases.
Arul, go ahead.
So as I had mentioned in my talk, we have established this parallel protocol to sequence pediatric oncology patients, and that IRB certainly took considerably longer to get approved because it's the pediatric population in general of having consent from the guardian and so forth. So there were other issues that have been involved, but I think one of the major challenges for us in the pediatric population is that even if we do find an actionable target in the pediatric population, oftentimes there will not be trials open for that drug, or that the dose hasn't been figured out for the pediatric population. So it's an even larger challenge in the pediatric population of actually matching the mutations to individual drugs, an even more significant challenge, I would say.
Thank you.
Oh, sorry. I'm not sure who was first, but maybe this person and that. I'm sorry, can you say your name?
Roy Jensen, University of Kansas.
Hi, Roy.
This question is probably going to fall to Jim here, but one of the themes that I've noticed today is that a lot of what we've been talking about is a commingling of traditional cutting-edge clinical care and a new type of clinical research that we haven't really defined yet. And because there are no current mechanisms to really support this activity. And a great example was just given by Pat, where they spent 3 hours at a tumor board, and I doubt if and I doubt if any of those went on a clinical trial, because you didn't have a trial for any of those patients, I bet. And so how in the questions to Jim, how is the NCI going to view this in terms of the center's program in fostering this kind of hybrid type of activity when in the past it has served you extraordinarily well to exclude any kind of dabbling in clinical care? And it looks like, we're about to smack into this brick wall here.
Okay, so
Jim, would you like to take that?
Yeah, we're still not going to dabble in clinical care, okay? But maybe I'll take this chance to describe 2 clinical trials, one of which you probably heard a little bit about at the Center's Director meeting a couple months ago. That's the so-called MATCH trial. That's a really innovative name. But it basically is a study that we hope will be open in about a year. And will involve screening somewhere between 4,000 and 5,000 patients with a specific set of actionable mutations and a panel that we've already worked out for the trial that I'll tell you about next. That will be open. The idea will be that we'll screen, as I say, upwards of 5,000 patients, fresh biopsies. Trial that will be open at all NCI-designated cancer centers, cooperative groups, and all the CCOPs. We had a meeting a week ago yesterday with representatives from 20 pharmaceutical companies who essentially to an individual agreed to, and they will be asked for formal written pledges to provide drugs that have phase 2 doses so that we will have a pharmacopoeia to be able to treat patients around the country who are eligible for this study. They had this trial. There will be a limited data set. Still, there will be collection of clinical data, but it will be relatively limited, not the same as a large cooperative group trial, but clearly there's sufficient data to interpret what it is we do. The idea is to set this up as basically a discontinuation trial. So patients who are not eligible will still have fresh biopsies, and those eventually will be studied with exomes and a very wide variety of other instruments. The patients who are eligible will go on, and those who get treated and respond will keep being treated. And when they progress, they'll get biopsied again, and we're trying to develop we have the resources to pay for that second biopsy. The patients who have stable disease will actually be treated. The initial thought is that they'll be treated for 6 months and then asked to take a drug holiday and to go back on again. So it won't be a formal randomized discontinuation trial, but at least we hope to learn something in that context. And the issues are, there are many issues. I think we have the money, just the biopsies alone are over $10 million for a study of this nature, but because we have to pay for them. But I can assure you that they will be done. At least they will be, the money will be available. It will be done, the mutational panel for eligibility with a 2-week turnaround will be done by a limited set of institutions who will have to compete to participate and will be QC'd and overseen by the lab that we set up in Frederick to do exactly this. And so I'm very encouraged because in answer, I don't know, just to answer Sandy, but to another question that came up about, I guess it was Pat's, about what's the best drug. Perhaps the most interesting, amongst the most interesting things that we learned a week ago Sunday with all of our pharma representatives is that they were actually already, after hearing about this, competing to make sure that their drug for their target, how would we randomize between the BMS drug and a Pfizer drug, right? And the idea is to have all of these drugs, as many drugs available, so that we can actually do, some, if not formal comparisons, at least have a variety of different cohorts to be able to use, because Pat is absolutely right, you really want to use amongst, if not the best, at least have a range of the best agents. So this study, while it will be run by a cooperative group, it will actually be run for the NCI by the new NCI Clinical Trials Network, available everywhere and done a little differently because it will be available at cancer centers. And so it's going to be very expensive enterprise, but I'm here to tell you that Dr. Varmus has committed to funding the tissue acquisition, funding the analyses, and funding the clinical trials costs across all of our 3,000 sites. So this is probably the best job that we can do to get a pharmacopoeia, which I know is going to happen now after a week ago, and ship those drugs where the patients are. So that we'll go on and to have a study that doesn't have an untoward, just an enormous burden of data collection, but still we'll have the sufficient data, hopefully, to understand the information. And I don't know that this will or will not develop enough information to provide information for any one of the companies to get an indication. It isn't really being set. I mean, it will be under an NCI IND/IDE, right? And we will do that. But whether it will be sufficient for an indication, I don't know. But I hope actually to learn how to do these trials at the end of the day. And the last thing I should say about this study, and I know I need to shut up in a minute, is that number one, not only the initial panel data, but everything else that we do it's a government study, so it will be available to the entire community, hopefully, because I think there will be in it, since we'll have tissue, fresh tissue, and especially the tissue off study at progression, when eventually those exomes get done or other kinds of things, it ought to be a marvelous resource for investigators around the country. I'll just say a very brief, if I may, thing about a trial that will start much sooner, something we call the MPACT trial. This is something that I've devoted over 3 years of my life to. And I really bring it up because it is, and the reason that there's no right or wrong, and that if we could do what Rich would like, it would be, God knows my life would be simpler, is it, and perhaps it's a good thing that Liz and Tony are off the line, okay? Because we submitted this trial after 2.5 years of work on basically Labor Day last year, right, to the FDA. And we're initially told that we wouldn't need an IDE. And then, of course, when they had, this is the first protocol they've ever seen like this that has an algorithm that specifies, it's all CLIA certified, done in a specific way. There is a formula, right? And this is one, if not the first, one of the first times they've actually had to react to an IND that had such a thing in it. And they certainly did react, about 25, I don't know how many pages of reaction, right? But it's okay, because that's what your tax dollars pays for. You know, for us to learn all of the things that a sister agency actually wants done. And then we responded, and now they reacted again, and we're in the third go-round. But we will get this open. This will be a smaller study, about 1,000 patients screened with 5-6 different drugs, a series of nested phase 2 trials, but it'll be a randomized trial of treatment based on the mutation versus treatment with the same group of drugs that you get treated, not matched. And then when you progress, you get what you should have gotten in the first place, and we'll find out, because it's powered so that we can ramp up all of the phase 2s to try to answer the question: does it make a difference? And again, I think that it's really important to remember what Pat has been saying, is that at least these two efforts, although they'll be very large, I don't think will answer the question per se. I don't think they should be expected to actually, because whether we chose exactly the best drug for the best mutation, whether it's ridiculous to think that a single agent is going to be useful, et cetera, I think it's much more important that we have spent the time and effort to find out what the regulatory agencies actually want us to do, so that all of you, because you will all have your own studies at your own cancer centers, I hope that it will be easier. Having jumped through these hoops, if that alone happens, and we have a public database of mutations and response and clinical data, I think the tax dollars will have been well spent.
You know, we have a similar study, but it's much smaller. We're only looking at one tumor type, and it's being partially, and I emphasize that, only partially funded with the money that Stand Up to Cancer and MRA gave us in the non-BRAF mutant patients, where we are bringing about 15 novel agents under one umbrella IND, and we've been preliminarily told that we don't have to file a device application. However, we're back at the FDA on the 15th of July, And I can tell you that these are enormous projects to do. He's lucky that multiple companies with the same target want to work with him, because that's been an area of challenge for me, because I know that I need drug combinations, and I know that I need different MEK inhibitors, because I know, based on preliminary data that's come out of the labs of Neil and Dave Salt and Neil Rosen that are on my Stand Up to Cancer, that we need these things. And it's very challenging just to get all the letters of cross-reference, because they're all, I'm very impressed. I can use him as, because a lot of the companies don't want to give you their MEK inhibitor if you have another MEK inhibitor. The only way I'm getting around it is because I know I need MEK AKT, I know I need MEK PI3K, I know I need MEK CDK, so I can partially get around that by looking at the combinations that I'm working through. But I've been working on this, how long, a year and a half? And I mean, we're just getting to the FDA on the 15th of July, and that's not to file our protocol, that's to go to our second Type B meeting. And there's a lot, they're very expensive trials. Yeah.
So that was great. I'm glad. I was going to ask you to comment on that. So I think that just it shows you the excitement in the academic community doing these sorts of approaches, and it sounds like we're going to need many different ways of really trying to get at the question of how genomics is going to impact on clinical care. You've been waiting patiently.
Yeah, thanks. Mike Bookman from the University of Arizona. I just, these are great innovative ideas, and it's really encouraging to see some of these approaches moving forward, even as difficult and as expensive as they might turn out to be. But I just wanted to be sure we don't forget biology. And just brief example is trying to understand how to use PARP inhibitors in the right patient population and evaluate the results in a meaningful way, so that we know that loss of germline or somatic BRCA1/2 function doesn't just make the tumor sensitive to a drug. It actually changes the biology of the disease. In several settings, those patients actually do better. They respond not only to specific targeted agents like PARP inhibitors, but they respond to other agents that are not specific, that damage DNA through other mechanisms related to DNA repair inhibition. And then there's about 40 other genes that can contribute or modulate the response to these agents, as well as the primary attention-getting mutation or loss of function of BRCA1/2. So having a well-designed prospective trial where these are used in combination with drugs or used as single agents, and being able to look at not only just genomic changes, but look at prognostic factors for the disease and outcomes, it's also important. And I hope we continue to be able to occasionally have some of these somewhat more conventional but well supported trials that ask the right scientific questions too.
Absolutely. Stan, are you, how are we, I know we're over time, so we have, of course, we could talk for another 3 hours, but as the chair.
Take one last question here,
Yes,
and we'll.
absolutely. Yes, ma'am.
I just have a quick question. Sharon Stack from Notre Dame Harper Cancer Research Institute. This all sounds tremendously exciting to patients that have the good fortune to live within the catchment area of an NCI comprehensive cancer center. What about the rest of the country that is requiring access through a community hospital setting? And it's a question and a comment moving forward. Please consider that population strongly as well.
So the big trial that I initially described, that will be open at all NCI-affiliated CCOPs. And I assume the networks of the comprehensive cancer centers as well.
So Lindsay, sorry.
Oh, that's okay. I was just going to say, from my perspective, this is the biggest ethical challenge, as well as how to ensure adequate access to cutting-edge science and care. So going forward, I would encourage everyone to consider this problem that we are insulated from being affiliated with a large academic medical center.
Lindsay and panelists, I want to thank you for taking the time, and everyone in the room who came from out of town, especially on a bit of a rainy day and have a ride or a flight back, for coming. We didn't design this to answer all the questions, but to raise them, which I think we successfully did, and hopefully we'll move the needle a little bit. And I hope we've generated enough discussion that some emails whiz back and forth between all of you. So I really want to appreciate the efforts of this panel, the moderators for the day, and all of you for attending. I need to make one more little announcement, and that is that university hospitals, is actually pretty proud of its new Seidman Cancer Center, so that those of you who might be interested in a quick 15-minute tour of the place before you head on home at the registration desk, we'll be getting a posse up to go over and have a look. And Nathan Levitan will be leading the tour in about 15 minutes. Thank you all for coming, attending, and again, feedback would be terrific.